I'm pleased to have the chance to present to you results of a site that's basically in your backyard. Let's see. So this is a site where there was an oil spill from a pipeline. It was at that time the Lakehead pipeline system. It now is operated by Enbridge Energy. It was Line 3, which has been in the news lately because they're proposing to replace Line 3, which has actually been the site of two of the largest terrestrial oil spills in the US history. And this spill was about 440,000 gallons. The biggest spill on Line 3 was 1.7 million gallons for reference. So this was about a quarter the size of the biggest spill on Line 3, but Line 3 has been problematic and is now getting old. So this is just an aerial photo of the spill in 1979 with the pipelines shown in black. This is a full-size tanker truck just for scale to give you a sense of how big this impact was. But fortunately, there were no waters of body nearby, so the oil didn't go into a lake or a river. It coated the tree. This is state forest land here. So in this talk, I'm going to start out by just showing you some cool pictures of the spill right after it happened and the initial emergency cleanup response. And then I'll have a few slides about the USGS study when it started and what the motivation for the study is. And then I'll show you some of the results. First about the spray zone, where oil just sprayed on the surface, and then the crude oil source where the oil had sank into the ground and forms a source of a groundwater plume. That's why it's called the source zone. And then I'll talk about the groundwater plume, and then the groundwater plume is now arrived at a lake that's about 340 meters down gradient. And so I'll cover some of the work documenting the plume arriving at the lake. So this line broke in August of 1979. This is where the break was. This is where the oil sprayed onto the surface, and then the oil kind of ran along the ground. And I'll show you some better pictures of the whole area that was impacted. The site's up here, located about 20 minutes from the town of Bemidji. What broke was actually a buried pipeline. It was under high pressure, so that's why there's a spray zone, because the pressure was high enough that it just blew off the overlying meter of soil and sprayed under pressure. They stopped it after 440,000 gallons had spilled. And the cause was a bad weld on the pipeline. So I have a picture of what a bad weld looks like. This is a 34-inch diameter pipeline. There's a seam welded along it, and that's what ruptured. So you can get a sense of, if a pipeline, how you might end up with a problem with the pipeline. So these are pictures of the emergency response. Here's where the pipeline and pipeline break was. So they've kind of dug it up to expose the oil-coated soil. They dug a big trench here to try and collect more of the oil. And, but some of that oil also infiltrated to the water table. This is the cleared pipeline right away. This is an old railroad that's gone now and is now a RTV trail and an ATV trail. This is the area where the oil sprayed, and then it kind of ran down here and ran and settled into some low areas here. This is behind this guy who's measuring oil levels while smoking. It was a different time then, I think. As where they scraped off the vegetation and burned it. So this is a common remedial tactic instead of burn the oil and the oil-coated vegetation. And here they're also pumping oil from a wetland that it flowed to. So overall, they were able to remove about 75% of the spilled oil. Pumping was about 80% of what they removed. And excavation and land farming accounted for about 6% of what they removed. And burning or evaporation accounted for about 10% of what they removed. So pumping was a major removal strategy, the thing that was most effective in removing the oil. So here's an overview of the site with these things illustrated schematically. So the black outlines are where the oil infiltrated to the water table and forms a source for groundwater contamination today. So there's a north, middle, and south oil pool. The yellow is where the oil sprayed onto the surface, and I'll be showing you some photos of that later. This is where the pipeline broke. The groundwater flow is towards this unnamed lake. These white dots are observation wells. So most of the studies are on the north oil pool and the north oil body groundwater contaminant plume. This lake is about 340 meters down gradient from the source. So for the USGS study, the motivation is because there have been documented more than 500,000 leaking underground storage tanks. These are mostly at old gas stations. So here's a tank being yanked from the ground, a leaky tank, and you can see that the liquid's leaking out of it as they're yanking it. At a lot of those sites, it's called a risk-based corrective action for is what governs the cleanup. So if it can be demonstrated that the fuel is biodegrading, then, and it's not threatening any receptors like a water supply well or a surface water body, then it's posing no risk and natural attenuation can be accepted. So we have argued that some kind of long-term study of the natural attenuation is important because tank sites still have contaminant remaining in the subsurface. Can you hear me okay?
Yes, we can hear you.
Good, okay, great. So our study is part of a program called the USGS Toxic Substances Hydrology Program. And the program, which began in '83, established sites of, that were contaminated with important categories of contaminants. So there was initially a wood treatment site, a low-level nuclear waste disposal site, a sewage disposal site. And so the program is still ongoing, and we are one of the two oldest sites. The other site still in operation is a sewage treatment site. So we study the fate and transport and natural attenuation of crude oil and refined, by analogy, refined petroleum fuels in the subsurface. And we provide the results to industry consultants, regulators, and educational institutions like yourself and research institutions. So this is a photo of the line of wells at the site and set up for our summer field campaign. So we have a trailer and a big, huge party tent to work under. So here's an overview of, again, the spray zone, the oil body, north oil body. This is the outline of the 10 microgram per liter benzene contour in 1996. And I'm going to be showing you results from studies of the spray zone, or some spray zone results, and oil body results and plume results. But the good news that I'll let you know right off the top is that the benzene plume has been pretty stable. So here was the 10 microgram per liter contour in 1996. And here in 2010, this is 3 microgram per liter benzene, so below the drinking water limit. And so the benzene plume has been stable. This is a direction of groundwater flow. And so here we're looking at a map of the water table contours. So the water table elevation in meters. The flow is perpendicular to the groundwater contours, and the observation wells are aligned in the direction of flow, so we can see the change in concentration as the groundwater contaminant plume migrates down gradient. The groundwater flow velocity is about 22 meters per year. So 5 years to go 100 meters, 10 years to go 200 meters. There's a wetland here that does have, that some oil did migrate to. So I'll just show you a cross section along this line. So this shows all the wells in the North Pool transect in cross section. So I'm not asking you to look at any particular wells, this is very small, but just a sense that we have wells at different depths, able to 6 to 8 meters below the land surface. And the 1996 benzene contour is right here shown in red. And that it's a sand and gravel outwash aquifer underlain by till. So it's very permeable, the lake that the plume has arrived at, and we're looking at more lately. So notice the well coverage is pretty thin down gradient towards the lake. This is our last well nest before the lake. So just give you, zoom in on this section here to give you a sense of the contaminant concentrations. So here's the land surface, 6 meters below the land surface to the water table, 8 meters below the land surface to the water table here. Section of flow. This is oil in the that's infiltrated into the aquifer. And this is kind of the outline of where the oil is greater than 10% of the pore space. So as the groundwater comes by, it dissolves contaminants from the oil and forms this contaminant plume that migrates with the groundwater. So these are 1995 contours. This is 5 milligrams per liter. So that's 1,000 times the drinking water standard for benzene. This is all of the BTEX compounds listed here, but mostly what is in the groundwater is benzene. But you can see that concentrations decrease by an order of magnitude in just 50 meters. So this aquifer is very bacteria to biodegrade the BTEX. And then above the water table, the vapors also come from the oil. So we have vapor concentrations, this is parts per million of air, total hydrocarbon vapors, but they also biodegrade in the unsaturated zone. So the vapors don't really make it to the land surface, even though it's only a 6 meter to 8 meter distance to the land surface. So also an effective bioreactor, aerobic bioreactor. So just to refresh your memory about biodegradation reactions, natural biodegradation reactions, they work because bacteria get energy, as we get energy from burning food with oxygen. Bacteria get energy from burning reduced carbon, in this case, the oil compounds, and converting it to CO2 and water. So if the oxygen is present, they will use that as they get far more energy from oxygen. But because there's so much oil in the subsurface, the oxygen is completely depleted. and they will use these anaerobic electron acceptors. At the Bemidji site, we don't have a lot of nitrate and sulfate, so the most important electron acceptor is iron oxides that are coating the aquifer sediments. When the iron oxides run out, then the only anaerobic reaction that's important is this carbon dioxide is listed as the electron acceptor. It's actually fermentation coupled to methanogenesis. So here is a schematic of the important processes at the site that is published in a paper by Professor Crystal Ng in the geology department there at University of Minnesota. Here's the oil coming from, it's present from land surface down to the water table. Within the oil, these N-alkanes, which are very insoluble, the bacteria that live on the edge of the oil and the water can biodegrade those to CO2 and methane. Those gases migrate towards the surface. The methane is actually biodegraded before reaching the surface by these bacteria called methanotrophs halfway to the surface. So this is only 6 meters from here to here. So halfway, 3 meters up, there's this bioreactor that degrades methane to CO2. At the water table, the groundwater flowing by dissolves the BTEX, and also entering the groundwater are non-volatile dissolved organic carbon compounds that are partial degradation products of compounds from the oil. So in the groundwater, those biodegrade. If iron is exhausted, they biodegrade under methanogenic conditions. Out here at the advancing front of the plume, where there is still iron oxidation process, is iron reduction. And what's most amazing is that even though my whole career, the focus has been on the groundwater and whether groundwater poses a risk to drinking water resources or the environment, 90% of the carbon that's biodegraded in this oil leaves via the surface, via the CO2 efflux. So first I'm going to talk to you about the spray zone. So what's really fascinating about the spray zone is that the oil that's coating the sediments has gone through cycles of wetting and drying to where the soil and the sand are now oil wet rather than water wet. And so the water just beads up on the surface of the soil like it does on a waxed car. And that hydrophobicity inhibits the plant growth because the water can't soak in. The plants can't get water to their roots. Thomas students a tour of the site, and it's just irresistible to play with these sediments because the water behaves so strangely. They dump their whole water bottle on here, and you can see what happens. So the ongoing research of this site is to look at whether these oil compounds that are on the sediments, we know that oil is photooxidized. We know that from the Deepwater Horizon spill because the oil sat on the sea surface. So those photooxidation products enter the water, they're leaching to the underlying groundwater. So we have a study of what those photooxidation products are and whether they form a health risk. And then John Niebuhr's professor there at University of Minnesota is studying how to remediate these soils, what you can add, for example, clays, to make these soils not be hydrophobic and allow plants to grow again. So next I'm going to talk about what has happened to the crude oil source. So this shows you how the crude, so here's the water table. The oil kind of migrated down. It doesn't go much below the water table because it's lighter than water, just like salad oil is lighter than water in salad dressing. As the water table is down lower and then gets entrapped, so that's why there's oil below, some oil below the water table, some above it. This is called a smear zone. These saturations are how much of the pore is filled with water. So 0% saturation means there's no oil in the pore. 60% means the pore has 60% oil and 40% water or air. This is all constrained by cores that were collected and laboriously analyzed for how much oil was in each section. So the oil source, this is a light paraffinic crude oil, so it's different from what comes from the tar sands in Canada today. It 3/4 of it consists of saturates, so that means only carbon and hydrogen. So the saturated compounds would be the aliphatics, which are straight chain alkanes and branch chain alkanes, and cycloalkanes. The aromatics form another 1/3, so that would be the benzene and the polyaromatic hydrocarbons cores. And then asphaltenes and resins are a small fraction. So each of those compounds has different fates, and that's why I'm pointing them out to you. So first we're going to talk about the aliphatics, the saturated hydrocarbons. And here are two gas chromatogram analyses of oil samples collected, one from the same spot, one in 1983 and one in 1999. The peaks represent, this is the time, elution time in the GC. So the shorter carbon compounds come out first, the longer compounds come out later. So this goes from 10 carbons up to 30 carbons. The tall peaks are the straight chain alkanes, which are highly degradable. These two labeled peaks and the lower ones are branch chains, which do not degrade well at all. And so you can see how much degradation has occurred between 83 and 99 by the fact that these branch chain peaks, pristane and phytane, are all that remain, and all of the straight chain alkanes have biodegraded in those 16 years. So that's a really effective statement of how much these particular compounds biodegrade. The biodegradation within the oil is methanogenic. So when they biodegrade, the bacteria produce methane. So here's the water table. The methane is measured in these places with the crosses. These are soil vapor samplers. So these are contours of methane concentration in percent of the soil gas. So in this case, 10% of the soil gas is now looking at some kind of flammable conditions down here with 20%. That methane migrates towards the surface. Oxygen comes down from the surface and the methane partly up. And then methane also enters the ground, is present at 5 mils. So the methane, instead of reaching the surface, what actually reaches the surface is CO2, which is a degradation product of the methane. We can measure CO2 at the surface with this instrument shown down on the lower left. Is a flux meter. It's placed over a collar, and you can see this blue collar under it. Shown above are the data bodies. So right here is the floating oil of the water table. The vertical axis is soil CO2 efflux in micromoles of CO2 per meter squared of area of the surface per second. So in the clean background where there's no oil, you can see that the bacteria and the degrading plant matter and the roots produce about 3 to 4 micrograms of CO2 per meter per second. Above the oil, you can see that the CO2 efflux is basically doubled up to almost 8. So the difference between the natural respiration and this higher value above the oil is called the contaminant soil respiration. And that can be measured to tell you how fast the oil is biodegrading based on how fast the CO2 is coming out of the surface. So now I'm going to switch to the aromatic fraction of the oil. So the aromatics are very soluble. Some of them are. And so what happens is the groundwater flows by these are oil-coated grains of sand. And the water flows through, it dissolves out the most soluble components in the oil. So in this case, these are meant to be little benzene rings. If there's a lot of oil coating the sediments, the water can't actually get in. And so less benzene kind of comes out. So if we look at areas in the oil body where there's not very much oil coating the sediments, a lot of the benzene is gone today. If we look at areas where there's a lot of oil still coating and the water hasn't been able to get through, there's still a lot more benzene left in the oil, in the source. And then there's something else that controls what happens is that some compounds than benzene. Ethylbenzene is about an order of magnitude less soluble than benzene. So when the water flows by, it doesn't dissolve very much ethylbenzene. So a lot of the ethylbenzene is still left in the oil. So I'll just show you how that translates into the data that we see. So the top plot is groundwater concentration versus distance from the oil. The bottom plot is how much benzene is in the oil compared to what the oil started with in 1979. So the bottom plot, we have, this is one oil sample. 1987, it had about 80% of the benzene was still present of what was originally spilled, 100% originally spilled. In 2008, only about 40% of the benzene is left in an oil sample that was collected in 2008. So the groundwater's been leaching out the benzene. And you can see how that's affected the groundwater plume. This is groundwater plume concentrations average between 87 and 95. And average values in the groundwater are about 5 milligrams per liter, about 1,000 times the drinking water concentration. By emissions are only about 2 milligrams per liter close to the oil body, kind of bounce around 2 to 3, migrated very much. So the benzene plume is getting lower in concentration and it's also stable. And it's getting lower in concentration because benzene is lost from the oil. Instead, the concentrations between 2007 and 1987 to 95 haven't really changed. So there's just as much coming out of the oil today as there was in the beginning. And the reason why is because it's so insoluble. You can see that the concentrations are an order of magnitude lower than the benzene concentrations. It's an order of magnitude less soluble. And compared to what was in the oil when it originally spilled, which would be 100%, we're still at close to 90% of what was in the original spilled oil. So these low solubility compounds are going to be in the oil for a while and forming a source of groundwater. We had just, we finished the oil loss, right? Did you guys get that slide?
Yes, I think we got the gist of it.
Okay, so now I'm going to talk about the groundwater plume. So this is a big schematic of the oil at the water table, the anaerobic groundwater plume, just to introduce this section. So this is the benzene data from 1988. You have 5 milligrams per liter benzene. So on the horizontal axis is distance, the vertical axis is elevation. So these are contours of concentration. By 2010, the benzene concentrations are lower because benzene is lower in the oil. The front, you might think this front is growing, but notice it in 1988, there were no wells out in front. So these wells that are out in front are new since 1988. So the benzene plume is pretty stable. In contrast, we have this stuff, we call it non-volatile dissolved organic carbon. So it's DOC, but it's not the volatile. So it's not BTEX, which are typically referred to as VOCs. And those are present in 20 milligrams per liter here in 88, 95. And then in 2010, we had values of like 50 milligrams per liter. And this front is migrating down gradient. So we have this 5 milligram per liter here, and here it's down to migrated to 200 meters down gradient. So this non-volatile dissolved organic carbon, this is not regulated by the EPA. And so our most recent efforts since 2016 have been concentrating on whether this, whether there's a health concern associated with this DOC that comes from the oil. And another reason why we're concentrating on it is here's concentration versus distance. And this is a 1995 concentration in red, and the plume is actually growing at 1 meter per year in over 20 years. And so the reason why the plume grows is because the electron acceptor is iron oxide. This on the left is a picture of a core collected from the aquifer. So you can see it's this nice sandy material. So it's a glacial outwash aquifer. You can see that the bottom of the core is kind of orangey colored. That's the iron oxide coatings. And we've got it. And then up higher here, that orange color has been stripped off because the bacteria that use the iron oxide are converting it to iron 2, which is soluble. So the shown here are simulation results by Professor Crystalline. At 7 years, 14 years, and 28 years, here's millimole per liter volume of iron oxide coatings. And by 28 years, you can see that the iron oxide coatings have been stripped for about 150 meters down gradient from the source. So everything up gradient, the only degradation reaction is methanogenesis coupled to fermentation. And this reactor capacity is sort of used up over time. So that's why the plume is expanding, but expanding very slowly at 1 meter a year. So we've been concentrating on whether there's any toxicity. Did I just lose you again?
Nope, we can hear you.
Okay, good. So I'm just going to show you some toxicity results very quickly. So we looked at 4 wells. These are wells within the hydrocarbon footprint where you still see BTEX and true hydrocarbons. These are wells where you only see partially oxidized hydrocarbons that are this NVDOC. And what we used is a calyx bioassay. Calyx stands for chemically activated luciferase gene expression. It's an in vitro method, so we're not testing live animals, we're testing cells. We used a human liver cell culture. The modified, so the cell line is transfected with a DNA construct with a luciferase reporter gene. The gene is under the control of what's called the hydrocarbon response element. And we know that that's associated with polyaromatic hydrocarbon response. And so we can compare the response of this mixture of DOC to the response of a known polyaromatic hydrocarbon that has health effects. And when this gene is replicated, it produces the luciferase light generating enzyme, which can be easily measured optically. So here are the results. I just have one result slide to show you. This is the NVD DOC concentration. And recall that the values are in here they're between 1 and 50 milligrams per liter is the aquifer values. And these are tests done at different concentrations. So this is, and the background well is the rust color. These are the wells in the plume. So the background, there's not much response until you get to 40 times higher than we actually have in the aquifer. So the native organic carbon generates a response, but only at 40 times the concentration that it's actually present at, which is 10 milligrams per liter. The response is highest at the wells closest to the oil, and lower as you move down gradient, although surprisingly 250 meters down gradient, you still get a pretty robust response at the measured in situ concentrations, which are about 3 to 5 milligrams per liter. So down, this is 10 milligrams per liter. So down in here are the actual in situ concentrations. But then these curves are compared to the PAH known standards to get a sense of how this relates. So, we're over time, I think I'm gonna maybe skip forward with the lake. I'll just skip to the lake result that the plume increases the bioconductance of the water because it produces CO2, which dissolves the sediment carbonates, increasing the carbonate alkalinity of the water. So the cool thing is that specific conductance can be measured geophysically. Here's the last well by the lake. It just shows you that the specific conductance plume has arrived at the lake shore. The background specific conductance is way down at 400. So these guys went out in 2018 and they conducted an electrical survey of electrical resistivity below the lake. So that's this instrument on the back of the kayak. And he's also carrying a ground penetrating radar to look at depth. And these data were actually, they took a probe and pushed it down below the lake, this lake bottom, pushed it down into the sediments. They got specific conductance. If it's green, that was like background values. They classified that as no plume. If it's red, it's elevated specific conductance, and they classified that as plume. So this was our ground truthing data set. This is the raw data from our kayaking survey. So you can see that how the tracks of the kayak ran, and you see high conductivity in location here, here, and here. And those roughly correspond to where we expect the groundwater contaminant plume to reach the lake from the north oil pool, the middle oil pool, and the south oil pool. So just sum up with that the long-term study, I spent a while talking to you about the oil source. It still contained, by 2010, still contained about 40% of the original benzene, but that the benzene is dropping, and so is the concentration in the groundwater plume. In 2020, about 50% total of the oil is biodegraded. So that's 40 years after the spill. The groundwater plume, the benzene plume is stable, so that's good news because benzene is the most toxic compound in oil. But the plume is dominated by this dissolved organic carbon that is partial transformations that come from the oil. That DOC plume is growing because the bioreactor is fueled by the iron oxyhydroxide coatings on sediment, and those get used up. Those partial transformation products, they activate a known hydrocarbon detoxification pathway in human liver cells at levels that would be comparable to a health concern from a polyaromatic hydrocarbon standard. So the next step in a study like this, these are in vitro, so they're just cells, and maybe the cells are just doing what they're supposed to. So the next step is to test zebrafish to see if the zebrafish embryos show any health effects. And then I showed you that the plume can be mapped under the lake by measuring electrical conductivity, and that's we're not measuring the hydrocarbons with those geophysical methods. We're measuring the specific conductance, which is a side effect of the biodegradation reactor. So that's the end of the talk. If we have time, I'd be happy to answer questions.
Great. Thanks so much, Barbara. We do have about 10 minutes. If there are folks who want to ask a question, you can just unmute, or you can also type a question in the chat if you prefer.
I'll try leaving my video on.
I got a question.
I have to get
Thanks,
special permission
Barbara.
to go to the office, but I think the next time I give a department seminar, I'm going to go to the office.
So thanks, Barbara. This is Carl Rosen. Appreciate you coming to the department for this seminar. Got a question about the microbial degradation. Is there any way to accelerate that with specific microbes, or you're just using naturally occurring microbes in the system?
You know, I think that it could be accelerated probably by adding sulfate as an electron acceptor. That's been demonstrated in the literature. And that would accelerate it in the source zone. One problem with sulfate is if you add it in the water, then the water just migrates. People have been adding gypsum, like they've been injecting these sort of gypsum hydrofracks into source zones, and that's been pretty effective in source zone remediation.
I guess you wouldn't be wanting to add nitrate. I guess that would help too.
Yeah, nitrate's not met with much, you know, enthusiasm from the regulatory community.
Yeah.
It's a contaminant.
Yeah, I can see that.
We've got this lake downstream that is not eutrophic.
Yeah. So one other quick question is, this is more general. There's a lot of news about putting more pipelines in. Is there any way that these types of spills can be averted with the newer technology? I mean, is this going to be an ongoing problem, or what's the?
Well, there's some nice statistics on the American Petroleum Institute website. They're a little out of date, but somebody did a study of pipeline spill frequency in 2010, and the frequency showed an impressive drop. So the pipeline industry, although they certainly still have spills, the frequency of spills has gone down over time. And that's, and it's really clear why they want to replace line 3, because has been responsible for a number of spills.
Yeah, exactly.
So just, yeah.
Thank you.
Someone asked, what are the future plans? I saw that. So I did mention we want to do in vivo studies, because in vitro studies are just a, their starting point to see if you need to do further studies for health effects. And what we're studying right now is the whole mixture of what's there, but what we'd like to know, there's a whole body of work that I didn't show you on what's in this DOC. And that work has just been published, and it indicates that maybe some polyaromatic cores with 3 or more polyaromatic rings that have oxygen functional groups on them. So that makes them soluble. Otherwise, they normally would not leave the oil because they're poorly soluble. But they are possibly in the down gradient plume and possibly not biodegrading based on the data that we have. So we're going to try to zero in on those polyaromatic. They're called oxy-polyaromatic hydrocarbons. So we're going to have a big focus on those coming up.
Hi, Barbara, this is Mindy. I'm wondering how the regulatory community is interacting with you and other researchers considering updates to rules and monitoring policies and that sort of thing.
Yeah, so the regulatory community is following this work on the dissolved organic carbon pretty closely, because there is previously there's been an argument that the polluter, the responsible party, should be held accountable for the true hydrocarbons, the compounds that originate in the oil. And this work is showing that there are these partial transformation products that are present in the aquifer. And so some regulators are concerned about regulating those partial transformation products, but they don't know how to regulate them. They don't know what to measure, and they don't know how to do a health risk analysis for them. So we're working very closely with the regulatory community to release our results as quickly as possible, and get some in vivo results, because in vitro results are not, they can't be used for calculating risk. Yeah.
Great, thank you.
And I guess I should also say there's a lot of interest in the source of natural attenuation. So how fast the oil is biodegrading. There are a lot of sites where these CO2 efflux techniques are being used, and it was developed at our site, but we want to show what happens in the long term, that today's CO2 efflux is not necessarily what you're going to measure 10 years from now, because I think it looks to us like the rate of oil degradation is slowing down, and that the oil's got a lot of things that don't degrade very well left in it. So there are groups that are watching that work pretty closely as well. Yeah.
Okay, are there any final questions for Barbara? All right, I don't hear any. I don't see anything else in the chat.
Great, well, thank you for your attention.
Yes, thank you so much, Barbara, for joining us. And Sorry about the technical issues, but I guess that's just the nature of the beast right now. So
Yeah.
um,
Interesting what you do with the recording.
yeah, we'll see what we can figure out. But thanks again. And thanks everyone for joining us. And we'll see you for our next seminar in a couple weeks.
Bye-bye.
Bye.